Good day to you from Inside Drug Discovery. This video describes the discovery program of MORF-627, an integrin alpha V beta 6 inhibitor for the treatment of selected fibrotic diseases. The video partially summarizes the discovery program as described in a Drug Annotations article from the Journal of Medicinal Chemistry by scientists at both Morphic Therapeutics and Schrodinger in the United States. A link to the article can be found in the video description. The article opens making a case for the unmet medical need for treatment of fibrotic diseases, specifically IPF, idiopathic pulmonary fibrosis. In the pathology of IPF, integrins, a family of proteins, activate and cause the release of a group of cytokines, TGF-beta. TGF stands for transforming growth factor. These cytokines bind to ALK5, activin receptor-like kinase 5, to initiate tissue fibrosis. Integrin alpha V beta 6 is highly expressed in lung tissue of IPF patients and has therefore emerged as a promising therapeutic target for treatment of IPF. Integrin alpha V beta 6 has three main conformations: bent closed, extended closed, and extended open. The extended open conformation is associated with TGF-beta activation and release, and ultimately initiation of the fibrotic response. The plan, therefore, was to design an inhibitor that binds specifically to the bent-closed conformation to minimize the fraction of alpha V beta 6 that is available in conformations that activate for TGF-beta. Conformational distribution changes in alpha V beta 6 can be measured through size exclusion chromatography. The targeting of integrins for fibrotic diseases is known in the literature, so much of the assay methodology has already been well established. Here are two literature compounds that bind various integrins. The top compound is known to bind the open conformation. One key feature is this tetrahydronaphthalene, which is positively charged. At physiological pH, the group binds an important aspartic acid residue found in integrins. Another key feature is the carboxylic acid. The anionic carboxylate binds a magnesium ion in the active site. The bottom compound is known to bind closed conformations of an integrin. The bottom compound also has positively and negatively charged groups, and its preference for the closed forms is attributed to this nitrogen, which is protonated and serves as a hydrogen bond donor specific to the closed conformation. As a spoiler alert, in compounds described in the rest of this video, we will see a lot of this tetrahydronaphthalene group, an acid on the opposite side of the structure, and an amino group close to the acid. Based on screening a small custom library, the researchers focused on compounds with the scaffold shown in the upper left. You can see the tetrahydronaphthalene system on the left, a carboxylic acid on the right, and a tether connecting the two. Inclusion of a phenyl group improved potency for the desired integrin alpha V beta 6. Selectivity against alpha V beta 1 was encouraging, but selectivity against alpha V beta 8 was poor. Some tweaks to the tether, replacing the amide with an oxymethylene, reduced potency for alpha V beta 8 and therefore improved selectivity. Compound 12 was found to bind the bent closed conformation of integrin alpha V beta 6. Furthermore, the MedChem team obtained a co-crystal of compound 12 and the binding domain of integrin alpha V beta 6. On the screen is the co-crystal of the ligand binding region of alpha V beta 6 bound to compound 12. Here is our ligand, compound 12. The rest of the structure in the screen is the integrin protein. Two key features are, one, the interaction of the tetrahydronaphthalene system with an aspartic acid residue, and two, the carboxylate of compound 12 binding a magnesium ion. The co-crystal confirms the expected target engagement of compound 12, but let's now focus on improving on-target potency. To improve potency based on information from the co-crystal, the researchers explored adding a small substituent, a cyclopropyl group, to the phenyl ring. Compound 12 is unsubstituted. All placements of the cyclopropyl group improved potency, but ortho substitution, as seen in compound 18, improved alpha V beta 6 potency to single-digit nanomolar levels. Admittedly, some selectivity over other integrins was sacrificed. Okay, that's enough for potency at the moment. Let's now think about ADME properties, specifically membrane permeability. With regard to membrane permeability, compounds like 18 pose a problem. The structure has 3 formal charges at pH 7.4. These charges diminish membrane permeability. Molecular modeling, however, predicts that structures like compound 18 with its long tether between groups can accommodate an internal complexation, an internal neutralization of sorts between the carboxylate and tetrahydronaphthalene groups. This protonated amine, however, remains as a barrier, so the researchers wanted to explore ways to reduce the basicity of this amine, reduce its extent of protonation at pH 7.4, make the overall molecule more lipophilic, and thereby increase the compound's membrane permeability. The apparent permeability of compound 18 was just 3.7 times 10 to the 6 centimeters per second. Placing a single fluorine on the pyrrolidine or aromatic ring raised apparent permeability to 15 and 12 times 10 to the 6th centimeters per second, respectively. Presumably, the inductive effect of the fluorine is reducing the basicity of the amine enough to raise permeability enough for a reasonable expectation of oral absorption. If you remember, addition of a cyclopropyl group in the ortho position of the aromatic ring gave a boost in potency with a loss in integrin selectivity. At this stage, the researchers explored larger ortho substituents to better tune the desired potency and selectivity. Compounds 30, 37, and 38 are listed here, but many other analogs were explored. The end result is that larger groups more fully filled a binding pocket, improved alpha V beta 6 potency, improved integrin selectivity, and generally improved membrane permeability. When inhibition of alpha V beta 6 was measured in the presence of human serum and therefore factoring in plasma protein binding, compound 38 showed the highest potency. Compound 38 was ultimately selected as the front runner for the program. Here are some PK parameters for compound 38, internally known as MORF-627. The PK parameters were determined across multiple preclinical species. The bottom row shows the predicted human PK values, with the predictions based on allometric scaling of the preclinical data. The predicted half-life was not listed in the paper, but it can be calculated from the clearance and volume and distribution values. Here is some evidence of preclinical efficacy for MORF-627. The disease model involves mice with lung fibrosis symptoms. The study monitored a downstream biomarker for an integrin alpha V beta 6 initiated pathway. The biomarker is PSMAD2. Higher PSMAD2 correlates to an increased risk of fibrosis. So the first column is dose, And the second column is response. The values in this table were manually read from a graph in the paper. Figure 12A if you want to look at the paper. So the PSMAD2 values shown here may not be exact. Regardless, MORF-627 does show a promising dose-response relationship. A higher dose of MORF-627 appears to afford lower levels of a fibrosis biomarker that is downstream of MORF-627's target. The study did include blood draws from the mice and monitoring of plasma levels of MORF-627. The researchers were able to model a sigmoidal relationship between response and the log of plasma concentration. The inflection point of the sigmoidal curve corresponds to an IC50 value of 8.3 nanomolar, which is close to the IC50 value of MORF-627 in cellular assays in the presence of human serum. This in vitro in vivo correlation helped to validate the broad design of the research program and assist with human equivalent dose predictions. In the process of performing toxicology studies to secure IND approval, researchers observed unfavorable outcomes in a 28-day oral study in monkeys. Dosed animals developed, quote, tumors in the urinary bladder consistent with urothelial carcinoma, unquote. The scientists at Morphic have published this outcome separately, and their findings can be seen through the URL at the bottom of the slide. I think it's worth noting that MORF-627 showed no signs of genotoxicity through early in vitro assays. Regardless, with the unfavorable toxicology results, the advancement of MORF-627 was terminated. Research into integrin inhibitors, however, continues at Morphic. That is the story of the drug discovery program for MORF-627, an integrin alpha V beta 6 inhibitor. The compound has high selectivity for alpha V beta 6 over other integrins, is orally available, and has a reasonable PK profile. Thank you for watching this video. If you have any comments or questions, please leave them below the video. Also consider liking the video or subscribing to the channel. Take care.